Hey everyone! In the last video, we saw how the cell cycle produces two cells starting with one cell. In this video, we'll look at mitosis and how the events of mitosis lead to the orderly sorting of chromosomes into those two daughter cells. Now, as always, remember to fill out the learning guide at the end of the video, and by the end, you should be able to answer two questions. And our two questions are: what events occur during each phase of mitosis? And how do these events ensure that each daughter cell gets two of each chromosome type and the same genotype as the parent cell? In the last video, you saw that M phase is the part of the cell cycle where the cell distributes two of each chromatid into each daughter cell. While the separation of the two cells is accomplished during cytokinesis, the events of mitosis are what ensure that each daughter cell gets two of each chromosome and ensures that each daughter cell gets the same two alleles as the parent cell had. The cell can't tell which chromosomes are chromosome 1s and which are chromosome 2s, for example. They can't distinguish different chromosome types from each other. So the cell is essentially blind to this information. So how does the cell evenly distribute the chromosomes into daughter cells without being able to determine which chromosome is which? It does this in much the same way two blind people would distribute pairs of something they couldn't see. Consider a variation of the blind man's riddle. Two blind men, who just also happen to be extremely talented musicians, are sent to the store to buy socks. Now, both men have partners that like blue, orange, and pink, so they each buy one pair of socks of these colors. Each pair of socks is held together by a band. While waiting for the bus, both men drop all of their pairs of socks so that the pairs get mixed up. There is no one at the bus stop to help them, but by the time the bus comes, each man has one pair of blue, one pair of orange, and one pair of pink socks in their respective shopping bags. So, how did they do it? The problem faced by the blind men is the same problem faced by the cell. How do you evenly sort pairs of something, pairs of chromosomes in the case of the cell, without being able to see them? The cell and the blind men solve this problem in the same way. First, I'll show you how the cell does it through mitosis, and then you can try to answer the blind man's riddle for yourself. Or I suppose you could cheat and use Google, but where's the fun in that? Most cells spend most of their time in interphase. Here are a few fava bean root cells in interphase. Notice that the individual chromosomes in the nucleus are not yet visible because they are not yet condensed. Following interphase, the cell enters mitosis. The phases of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. Mitosis is a continuous process. We just use these words to describe different parts of it. First, we'll look at prophase. When the cell goes from G2 into prophase, the chromosomes condense greatly and can actually be seen with a light microscope. Here's a fava bean cell in prophase. You can now see its individual chromosomes. Also notice that the chromosomes are still contained within its nucleus. Also, during prophase, the mitotic spindle or spindle apparatus starts to form. The mitotic spindle is the collection of structures that move chromosomes during mitosis. It includes the microtubules, shown by these pink lines, made of tubulin. It also includes proteins associated with the microtubules, including the motor proteins, shown here by these blue dots, that in a later phase generate the force on the spindle fibers to move the chromosomes. The mitotic spindle also includes the centrosomes, shown here by yellow circles. The centrosomes are where the microtubules are first formed. As you'll see, the location and size of the mitotic spindle will change greatly throughout mitosis. In animals, the centrosome is made of two centrioles, shown here. During prophase, these mitotic spindles move towards the ends of the cell. Next is prometaphase. This is when the nucleus dissolves, allowing the growing microtubules to bind to the kinetochores of each sister chromatid. Each sister chromatid is attached to a microtubule from different centrosomes, from different sides of the cell. The microtubules that attach to kinetochores of sister chromatids are called kinetochore microtubules. Okay, maybe not the most creative name in the world, but that's what they're called. The end that is attached to the kinetochore is called the plus end, and the end that's attached to the centrosome is called the minus end. Other microtubules do not attach to a kinetochore, but instead bind and overlap with a microtubule from the opposite side of the cell. These are called polar microtubules. A third type are the astral microtubules, which connect the centrosome to the cell membrane at each end of the cell. Next is metaphase, when the microtubules get shorter and longer, moving the chromosomes to a region near the middle of the cell called the metaphase plate. You can see this as well in a fava bean cell in metaphase. Next is anaphase. Here, the cohesin proteins that hold the sister chromatids together are broken down, and the two sister chromatids are pulled apart. In these two fava bean cells, we can see the chromosomes have moved towards the ends of the cell. This one is in relatively early anaphase, and this one is in relatively late anaphase. But where does the force to pull these chromatids apart come from? There is still a lot we don't yet know about how chromosomes are moved during mitosis. What I'm going to show you next will differ a bit among species and is certainly incomplete, but we won't let that stop us from covering this interesting process. Anaphase has two parts, anaphase A and anaphase B. Here we will just follow the movement of the two chromatids of one chromosome. The forces that move the chromosome are different in each part. During anaphase A, the tubulin subunits are removed from the kinetochore microtubules, making the kinetochore microtubules shorter, separating the sister chromatids from each other, and pulling these chromosomes toward the centrosomes. This is accomplished by either disassembling the microtubule at the plus end or the minus end, or in some species and cell types, at both ends. At this point, follow the first link below this video to view a quick animation of a microtubule being disassembled. It also shows one being built. I'll give you a minute to pause the video and check it out. Okay, so during anaphase B, the centrosomes and the chromosomes connected to them through their microtubules are both pushed and pulled towards the ends of the cell. The centrosomes are pushed towards the ends of the cell when the overlapping polar microtubules push away from each other. The movement of the centrosomes towards the ends of the cell pulls on the kinetochore spindle fibers, or microtubules, which in turn pulls the chromatids. At the same time, each centrosome is also pulled towards its end of the cell by the shortening of the astral microtubules. So it's these that get shorter. All of this, the shortening of the kinetochore microtubules, the pushing of polar microtubules against each other, and the shortening of astral microtubules, results in the chromatids moving towards the ends or the poles of the cell. The force behind these movements is often produced by proteins called motor proteins. Just as the motor of a car provides the force to move it, these motor proteins provide the power to move chromatids. For example, in the polar microtubules, motor proteins walk towards the positive ends of the microtubules, pushing the microtubules and the centrosomes that they are attached to away from each other. Now, next up, we have telophase. During telophase, a nucleus forms around each group of separated chromosomes, and the chromosomes decondense. In our fava bean cell, we see the nuclei have reformed, and that the chromosomes are no longer condensed or visible. After cytokinesis, this cell will become two separate cells. Now that we've completed mitosis, let's take a look at all the phases in the fava bean root cells. Here's prophase, where the chromosomes condense and become visible; metaphase, where the chromosomes line up at the metaphase plate; anaphase, where the sister chromatids separate; and telophase, where the nucleus reforms around the chromosomes and they decondense. At this point, I'd like you to check out a short clip of real cells going through mitosis. In the clip, the DNA is fluorescing red and the microtubules are green. I'll give you a moment to pause the video and click on the link under the video. Mitosis must produce daughter cells that have the same genotype. As you'll see, evolution has produced a simple and elegant way for cells undergoing mitosis to accomplish this. In contrast, meiosis I can produce daughter cells with different genotypes. To see why this is, let's follow 3 genes on 3 different chromosomes in cells that undergo either mitosis or meiosis I. In this example, the parent cell in both is heterozygous at all 3 genes. One reason for this is a difference in how sister chromatids are moved during mitosis and meiosis I. During mitotic metaphase, individual chromosomes line up at the metaphase plate. These are replicated chromosomes, so the sister chromatids of each chromosome have the same alleles. When the sister chromatids are pulled apart, they go in different directions and ultimately into different daughter cells. So each daughter cell gets one allele from each of the replicated chromosomes. For example, because each sister chromatid of this chromosome is pulled in a different direction, one big A and one small a will go to one side of the cell and eventually into the same daughter cell. And the other big A and the other small a will go into the other daughter cell, giving 2 daughter cells with the same genotype, big A, small a. The same thing goes for gene B and gene C. But during meiosis I, homologous pairs of chromosomes line up at the metaphase plate. During anaphase I, the sister chromatids stay together. So 2 of the same allele might get pulled to one side of the cell and subsequently inherited by one daughter cell. And 2 of the other allele would be inherited by the other daughter cell. In this case, after meiosis I, one daughter cell would be big A, and one would be small a. The same thing could happen for genes B and C. They can have different genotypes because the sister chromatids were not separated during anaphase I. Which chromosome and alleles a meiosis I daughter cell gets depends on how the chromosomes line up during metaphase I and on which chromosome is bound by a microtubule from which end of the cell. If a microtubule from this end of the cell happens to grab these 3 chromosomes, then one of the daughter cells will get the alleles on those chromosomes, and the other daughter cell will get the alleles from the other 3 chromosomes. Okay, so let's do some chromosome counting as we go through mitosis. At the end of G2, the cell has 2 of each chromosome type, for a total of 6 chromosomes, 4 copies of each gene, and in this 2n=6 species, 12 DNA molecules because they were replicated during S phase. The chromosomes condense during prophase, are bound by microtubules during prometaphase, and move to the metaphase plate during metaphase, but the number of chromosomes, gene copies, and DNA molecules doesn't change. During anaphase, the chromosome number doubles as they are pulled apart because each DNA molecule is now its own chromosome, so there are now 12 chromosomes. But the number of gene copies and DNA molecules doesn't change. These numbers don't change during telophase. The chromosomes just decondense and get surrounded by a nucleus. After cytokinesis, the cell will split into 2 cells, and each daughter cell will have 6 chromosomes, 2 copies of each gene, and 6 DNA molecules. Now that we've described how cells are able to give 2 of each chromosome type to their 2 daughter cells, pause the video and see if you can solve the blind man's riddle that we discussed before. Good luck! Okay, so by this point you should be able to explain what happens during each phase of mitosis and how the events of these phases lead to the production of daughter cells with the same genotype. If you don't feel like you can yet, feel free to watch part or all of the video again, and remember to fill out the learning guide. Now in the next video, we'll take a look at the not so good things that can happen when chromosome movement during mitosis goes wrong. See you then.